something.
Yeah. I'll buy you one of those.
Yeah. Because, like, what's the point of making videos if you can't understand?
Remember the phosphorylation thingies?
Where is it?
You passed it. Go right there.
Okay, thank you. All right, so ATP in living systems, 90% of the ATP is made through osmosis. This is just the movement of really it's going to be hydrogen ions across a membrane. That concentration gradient of the hydrogen ions is going to be what powers ATP production. This is going to occur in the mitochondria, chloroplast, and the plasma membrane of aerobic prokaryotes. So you see here we've got the mitochondria, which used to be a prokaryote, hypothesized. The matrix is on the inside. We've got the intermembrane space. We've got the inner and outer membrane. ATP synthase and electron transport chain are in the inner membrane. And essentially what they do is the electron transport chain pumps a lot of hydrogen ions into that inner membrane space, builds up that concentration gradient, and then that is used kind of like the water behind a dam to drive ATP production. Metabolic pathways involved in cellular respiration, there's 3. Glycolysis, oxidation of pyruvate, and the citric acid cycle. And the third one is oxidative phosphorylation. So in AP bio books, they have really complicated names for them. In the regular biology book, it's glycolysis, the Krebs cycle, the electron transport chain. Okay. But as with all things in AP bio, it makes it more complicated. I was actually looking at a presentation for this, and it had a picture on there, and it's like you thought the mitochondria was the powerhouse of the cell. No, it's actually the site of oxidative phosphorylation.
Unity on the key.
So remember, going back, oxidation, oil rig. Oxidation is what?
Oil. No, oxygen.
Okay.
No,
Oxidation is loss of electrons.
Yes.
Okay. Oil rig, reduction is gain of electrons. So this tells us if oxidation of pyruvate, what's happening to pyruvate? It is losing electrons. What's happening here?
Also losing electrons.
Losing electrons. All right. Very good. And then you've got that formula that you should all have memorized.
Yeah, not yet.
C6.
Yeah, not
You should have it all memorized.
Glucose,
Yeah.
oxygen, carbon.
All right, explain why it's more metabolically efficient for cells to extract energy from ATP rather than the bonds of carbohydrates directly.
Isn't
What are your thoughts?
ATP just always ready to energize?
Yeah.
ATP is a short term.
There you go, perfect, right? ATP is always ready. It's very short term. It packages energy in really small packages that can be used by proteins to get work done. Carbohydrates are large energy that you have to do some work to get that energy out of, right? So ATP is short, really quick energy. On the cellular level, carbohydrates, it's more like, all right, this is the gas station, and then the ATP is the gas tank in your car. Okay, that's kind of the analogy that we use in the regular biology group. Second questions. What's the importance of electrons for the transfer of energy in living systems? What's your thoughts on this one?
Oh, because consumers can't make their own energy, but producers can, right?
Yeah. But let's look at how do we make energy in cellular respiration, right? We make ATP. We make a lot of energy here. What's happening in oxidation?
Losing electrons.
Yeah, it's the movement of electrons, right? And so these processes, the movement of electrons, specifically the movement of high energy electrons, are actually what drive this. So when we talk about cellular respiration, that's why the movement of electrons is so important, is because they are usually going to be the ones carrying the energy that will eventually be used to make this. Okay? Make sense? A little bit? How is ATP used by the cell as an energy source?
No.
Yeah, it's consumed. What does it turn into?
ADP.
What do you say?
ADP.
ADP, adenosine diphosphate. So this is where we call phosphorylation, where the phosphor group, that phosphate group from ATP, will be attached to a protein, and then that is how it transfers that energy, by moving that phosphate group. Okay. All right. Any questions? Yeah. We're done with 7.1, guys. Aren't you excited?
Yay.
Now 7.2, glycolysis. What does glycolysis mean?
Makes
The
glucose.
breakdown of
Just
Breaking
separation.
down of glucose, splitting glucose. Perfect. Alright, so some of the questions we want to be able to answer when we're done is what is the overall result in terms of molecules produced in the breakdown of glucose by glycolysis? Yeah, it's 2 of each to the net. Okay. So glycolysis is the first metabolic pathway of the glucose metabolism. It includes 10 enzymatic reactions. An enzymatic reaction is just a reaction that uses an enzyme. Nearly all organisms and living things perform glycolysis. Has anybody ever eaten yogurt?
Oh yeah.
Okay, there's a bacteria called Lactobacillus acidophilus. It uses glucose as part of its energy making process. Same thing, anybody heard of tetanus?
Yeah.
Right, there's a bacteria called Clostridium tetani that is anaerobic. Y'all know what anaerobic means?
Built.
Doesn't mean-
Is it
You're
not?
thinking anabolic. Anaerobic.
Dang, you got me.
Anaerobic is the opposite of aerobic.
Breaks it down.
What?
Breaks down.
If you do aerobic exercise, what do?
Water exercise. Wait.
No.
Is it?
That's hydroaerobics.
Exercise.
No.
Oh,
It loses the oxygen.
I did this.
So anaerobic is without oxygen.
Oh.
Aerobic is with oxygen. So glycolysis is what would be called an anaerobic process because you don't have to have oxygen to get it done. Glycolysis is going to happen in the cytoplasm. O2 is not required, therefore it is anaerobic. Back on the topic of tetanus, tetanus is an anaerobic bacteria. It doesn't need oxygen. In fact, it doesn't like oxygen. So usually if you get a cut and worried about tetanus, the main concern is if it's a deep cut where it can be isolated from the oxygen in the atmosphere.
It's not oxygen.
Say what?
It's not oxygen.
Yes, ma'am. O2 with oxygen.
2.
Alright, glycolysis. It breaks down glucose with an enzyme called pyruvate kinase. It's going to release 2 ATP and 2 NADH. So we talked about that main question for the section. What are the products? We make 2 ATP, 2 NADH, and 2 pyruvates. The reason, why do you think ATP is important?
Short-term memory, or not memory.
Yeah, short-term energy. That's the goal of cellular respiration, is to make energy. Yes, sir. Who's at the door?
Then somebody walked by. I don't know if you guys got the word, but somebody walked by.
I heard someone knock. I heard a very light knock.
Sometimes what happens since there's that little fan out there, if the wind blows by, it'll like shut this door a little bit.
It wasn't that. I heard like a very light.
A very light knock.
I don't know.
Well, they're not important at
Might have
all.
somebody missing, like, doing this.
That could be. Yeah. That's okay.
So what are the overall products of glycolysis?
2 ATP, 2 NADH, and 2 pyruvates.
Yeah, very good.
Then you have 4. Dang, Mike.
Alright, so the inputs, 1 glucose, 2 NAD+, 2 ATP, and 4 ADP. We'll get into this in here in a second. Outputs are 2 pyruvate, 2 NADH, 4 ATP, and 2 ADP.
But 2 ATP are used, so net gain is
That's it. That is it. Alright, so this is actually a picture from the on-level biology course. So you can see here, we take 2 ATP to get glycolysis done, but we get out 4 ATP, so we have net gain of 2. And then we also release 2 NADH. These are going to be electron carriers. Their electron carriers are important because they're the ones doing a lot of the transfer of the energy in cellular respiration. We also end up with 2 pyruvate. How many carbons does pyruvate have?
6.
No. How many carbons does glucose have?
6.
Glucose has 6.
Okay.
If we break a glucose into 2 pyruvate, how many carbons does pyruvate have?
3.
3.
Yeah.
There you go. Okay, this is another visual of glycolysis where we have glucose. It is split by adding ATP into 2 pyruvates. We get 1 NADH per pyruvate molecule, and then 2 ATPs per pyruvate molecule as well. Okay. For a net total, we use 2 up here, we make 4 down here, so we get 2 altogether.
Yeah.
This is actually the step pathway for glycolysis in your book. First half of glycolysis involves 5 enzymes. It uses 2 ATP molecules. the book doesn't go in depth into what each enzyme is, so I am not going to make you memorize it, but I'm going to tell you that I had to memorize it in college, and it was terrible. So glucose is phosphorylated twice. What does phosphorylated mean?
Turn into 80.
No.
Don't you have to
Partially. I mean, you're on the right path, right? Phosphorylated means you add a phosphate group, okay? And then split into 2 to 3 carbon molecules called glyceraldehyde 3-phosphate. So we've got glucose, we phosphorylate it, add basically phosphate groups to it. Excuse me. And then we break it down with these various enzymes until we get this 3-glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. We really just call it 2,3-GP, G3P. So this is what we call this one, 3GP. And that is just the first step listed here. So in between here and here, even though in this picture it shows it as 1 step, it's actually 5. So just kind of, when we look at the overall picture, it looks really, really simple, but it's actually much more complicated than that. Okay? But as you can see here, we phosphorylate it twice.
So is phosphorylation like from ADP to ATP?
Yes.
Phosphorylated.
That's where I remember it from.
Yeah. Dephosphorylation is where you go from ATP to ADP.
Yeah.
Okay. Make sense?
I remember saying something.
All right. Second half of glycolysis involves phosphorylation without ATP investment and produces 2 NADH and 4 ATP molecules to generate 2 pyruvate molecules. So net production of glycolysis is 2 ATP. These are the only ATP produced in, I think that's supposed to say glycolysis, glucose metabolism by anaerobic cells. Okay, I need to fix this.
Don't press it.
You see what I did there?
What?
He tried to use his computer.
Oh.
Yeah, and it was not working. All right, so we've got the second half of glycolysis. First half generated is G3P. We have phosphorylation without ATP, and it makes NADH, and this is per pyruvate. We go through a lot of steps. We get out 1 ATP, 2 ATP per single pyruvate molecule, for a total of 2 ATP per pyruvate molecule, and then we get 1 NADH per pyruvate molecule. So the whole thing kind of looks like this. I don't think I'm going to have you memorize all the steps.
No.
Because here's the thing, like, I've memorized all the steps 3 times and I still don't remember them. So if I can't remember it after 3 times, then I don't think you should on that by the class, right? But you need to know, I said that, you need to know the inputs and the outputs and some of the intermediaries, right? You need to know glucose, you need to know 3GP, you need to know pyruvate. Okay? You need to know roughly where you input ATP and where you output ATP. You input 2 ATPs, you output 2 ATPs down here. Okay. And then you get out NADH as well. Is everybody dragging today? My
Yes.
4th hour is just like, and then I gave them 15 minutes to work on their own. And you know what they did?
They slept.
They didn't work, they talked. So guess what I did?
They
Did
were
more work? Did more work. All right, cool. We're gonna do more notes. I've got 20. Let's go. Let's go. All right. Outcomes of glycolysis. Come on.
Don't press execute.
Kate Holland, please come see Miss Amy.
That's not good.
Hey, nah, none of that in my class. All right. So what are the outcomes of glycolysis? What do we get out of it?
ATP.
Hey, how many?
2.
2 net
Yeah, 2 nights.
Okay,
but
we made 4, but we had to put 2 in.
Pyruvate,
How many?
2,
2?
and ADH.
How many? 2. Alright.
Yeah.
Alright. Start with glucose. We end with 2 pyruvate molecules, 4 ATP molecules, 2 molecules of NADH, but 2 molecules were used in the first half, so it's net negative 2. Pyruvate?
It was in Seagato, I don't remember what it was.
I remember it being a secret but I don't.
Yeah, I remember it.
I didn't do my Seagato until like 8:30 last night.
Alright, so glycolysis breaks down glucose, it's got 6 carbons, into 2 pyruvates. Pyruvates have 3 carbons. Basically, each pyruvate is half of a glucose.
So it's just half.
Yep, half glucose, right? This is kind of the process that it goes through to get there. 6 carbon molecule to 2 3 carbon molecules. What's this one called?
That's.
G3P?
Yeah, G3.
G3P. Okay. Glyceraldehyde 3-phosphate. You want to get super technical. Yeah, right. Yes, sir.
How do they split glucose? It would be like, so like in the equation, there's I think 6 carbons. They should put 3. Like so it goes 3 and 3.
Pretty much. So we phosphorylate it on each side, and then that causes it to break apart here in the middle. Okay. Does that get what you need?
Yeah.
Okay. Now, the interesting thing about sugars is they can be a ring or they can be a straight chain. Once we add the phosphate group to it, it's now a straight chain. And here it actually has it color-coded which carbons go which direction. So we have G3P, and then this is dihydroxyacid phosphate. Do you know what these double arrows mean?
Isn't that reversible?
Yeah, reversible. It can be any one, right? And so usually it will go this direction, specifically because you're going to lose a lot of this, because it's going to go to the Krebs cycle or the citric acid cycle, which causes this to become that G3P again. So does that answer your question?
Yes, sir.
Video?
We got time. It's loud. I'm going to speed it up because it sounds like it's just slow.
is the key energy molecule for all biological organisms. One source of ATP is glycolysis, a series of reactions that break down sugars while producing ATP. Glycolysis is a series of 10 reactions that convert sugars, like glucose, into 3 carbon molecules called pyruvate. Glycolysis is an important pathway for cells because it produces ATP and other products such as NADH and pyruvate that can be used later to produce more ATP for a cell. This key process takes place in the cytosol of the cell. Let's take a look at an overview of the key steps of glycolysis and focus on input and output molecules. The first 5 steps of glycolysis convert a 6-carbon sugar, glucose, into 2 3-carbon sugars, glyceraldehyde 3-phosphate. To do so, ATP is consumed in the first and third steps. Both of these chemical reactions are catalyzed by enzymes and kinases, and both reactions are irreversible. The next 5 steps of glycolysis produce energy. 1 ATP is made per glyceraldehyde 3-phosphate in both reactions 7 and 10. This means a total of 4 ATPs are produced in glycolysis. Both of these steps are carried out by a kinase reaction. Note that step 7 is reversible, while step 10 is not. It is important to remember that the products of steps 6 through 10 are formed for each of the glyceraldehyde 3-phosphate molecules generated in steps 4 and 5. This means that a total of 2 NADH and 4 ATPs are given in glycolysis. Because 2 ATPs were spent in the early steps of the pathway, glycolysis results in a net of 2 ATPs. There is one more key product of glycolysis to consider called NADH. NADH is formed in a 6th reaction by an enzyme called a dehydrogenase. NADH is an electron carrier with potential to produce more ATPs through a process called oxidative phosphorylation. Details of that process are shown in a separate animation. As you can see, glycolysis is a process involving multiple enzymatic reactions that break down sugars. Its major contribution to biological organisms is the production of ATP, which is used to drive many other biochemical pathways.
Crazy.
For real. Alright, practice question. What is the general formula for cellular respiration, and what role do oxygen and carbon dioxide
C6H12 plus 6O2
Okay.
yields 6H2O plus, okay, I gotta look now. 36 ATP. That's 36
6H2O plus 6 CO2
plus, oh, I have that now.
plus 36
ATP.
ATP. Okay.
I got the first part down.
We actually haven't gotten into what oxygen and carbon dioxide play into the process. We'll get to that when we talk about the Krebs cycle and the electron transport chain. Yeah. Oh, it was a multiple choice.
Mr. Rowell.
Here we go. All right, is it A, B, C, or D?
Six.
No,
Oh, see, no six.
there's no coefficient.
Yeah, there's no coefficients.
What's it say here? General formula. We'll do it without the coefficients.
It says
I'll be honest with you, I'm not sure if there's a right answer up here.
The one that was
C.
most right.
Oh, no.
There's two that are the same. Energy versus
Yes.
and
Taxes were
It's
due
A or
last
C.
time.
it's A or C.
Okay, here's the thing. Look at where your reaction arrow is on C.
Oh yeah, okay, then
Oh,
it's
okay.
A. I was right the first time.
Yeah, C6H12O6 plus O2 yields CO2 plus H2O plus energy, where glucose is oxidized to release carbon dioxide, along with energy, and oxygen is the final accepted electrons. Now, when we start talking about reading questions, you know, what did I say here? I said,
The general.
no, before we got that, I was like, we haven't got to this.
Yeah.
Right? Now, here's the thing. On this question, can you answer it without knowing the rest?
Yeah.
If you know the equation for cell respiration, you don't need to know the rest of this stuff, right? Okay. So that's something that I've had a couple students say they're having issues with these complex questions. Is like, you don't need to answer the whole question. You need to find the part of it that you know, break it down into different pieces, and then like do process of elimination. Because I think a lot of you are getting shell-shocked when you see a giant question. You're like, oh no, I don't know all of this stuff. You don't have to know all this stuff. You just have to know the pieces and be able to plug and play where they go. Does that kind of make sense?
Yeah.
Now, here's the thing. You're going to have to practice doing that because I think y'all have never had questions like that before where you've had to break it down in multiple parts and be like, all right, this part doesn't matter to me. This is the part I know. And granted, sometimes you have to know all three parts of the question. Sometimes you have to know one. But if you know one, what can you do?
Find elimination.
You can eliminate some of them, and then your chances go from being 1 and 4 to 1 and 3 to maybe 1 and 2, right? So when you're going through these practice questions for Chapter 7, the review questions, I don't want you to just be like, oh, I just need to get these done and get these right. Go through the process. Use whatever tools you have available, your notes, your books, the internet, to go through it as a process. Now, don't be like, paste the question in, like, oh, it's this one, click on it and move to the next one. It could be something as simple as, I'll use the example, there's two students actually that had the question. It was on this last test. Which process of the cell theory is most applicable to hospitals using sterilization and disinfectants? Okay.
I remember that.
Okay. Most of it was. The cell theory is all living things are made of cells. The cell is the basic unit of life. All cells come from pre-existing cells. Sterilization and disinfection has to do with killing bacteria. All right, so that's kind of the next step in the process. You have to know what sterilization and disinfection means. You have to know the processes of cell theory, and then which one of those are the most applicable to sterilization and disinfection. All cells come from pre-existing cells, right?
I didn't get that one right.
Okay, right. But just know, like, it's a step process, and y'all aren't used to taking it step by step by step. I think y'all are used to just basically saying, what is cell theory, and then having answer options, right? But it's like, now you have to know that and then be able to apply it in some way, shape, or form, right? So when you go through those review questions, especially the ones that are review or test prep questions, go through the process because that is going to help you more than anything, right? Because, like, I think I'm not so sure the knowledge is your biggest hurdle or if it's understanding and answering complex questions. Does that make sense?
Yeah.
Yeah.
Which one do you feel it is?
The second one.
Yeah.
The second one? Okay, so when you go through those, do the process. Because listen, I'm going to go ahead and tell you right now, you can Google every single one of those and the answer will be online. Every single one. Do I want you to do that?
No.
Definitely not at first, okay? See if you can answer it on your own. If you can't, okay, let me go to my notes, let me go to the book. Okay, There's pieces of it I don't understand. Let me figure out what sterilization means. Let me figure out what cell theory is. Let me figure out what oxidation is, what phosphorylation is. All right, now that I know this piece of it, can I make it into the big puzzle? I'm like, all right, well, I think it's this one because of this, and this. Then you can go check online. Okay? Like, hey, did I get it right? Yes, no, maybe.
So what I did on the other ones, like I would click it and then be like, I'm gonna make sure this is the right person.
Yeah, and that's fine to use it because, listen,
or
remember.
if you run out of time, just go through.
Yeah, that's on you. That's on you. Because here's the deal. If those of you that had me last year, what did I say? If it goes home, what should the grade be? 100. Okay? If it goes home, it should be 100. All right, use whatever resources you have available to you to get a good grade. If you want to work together in groups, I don't care. But you're doing yourself a disservice if you don't go through the process. Like, all right, how do I solve this problem? Does that make sense? Okay. Glycolysis occurs in cytoplasm of nearly every cell. Organisms from small circular colonies of bacteria, pictured here, and ignore that piece, to human only feature disc, perform glycolysis using the same 10 enzymes. Because of this, it's thought that glycolysis must have evolved in the earliest forms of life. ATP and energy is needed for glycolysis, representing the illustration provided, which is just the steps of glycolysis. Explain how this ATP gets its debt. The debt is paid off during the reaction. Okay, we think it's A, by the phosphorylation of fructose 6-phosphate, the oxidation of glyceraldehyde 3-phosphate, the formation of 3-phosphoglycerate, or by the formation of phosphoenol pyruvate. Let's go back here. All right, so we've got this 10 step. Where do we get ATP back? So if you were asked this on an AP exam, it would give you this. Okay. Right? And then you have to say, all right, where do we get ATP? We get it here when we make this, and we get it here when we make this. Okay? So we go from PEP to pyruvate, and when we go from bisphosphoglycerate to 3-phosphoglycerate. Let's see if those are options, huh? Okay. All right, yeah, by the formation of 3-phosphoglycerate. I believe that's right, yeah. Yeah, 3-phosphoglycerate. When we make that, we can make ATP. Okay, can I follow?
Yes.
Sweet. Nearly all organisms on Earth carry out some form of glycolysis. How does the fact support or not support the assertion that glycolysis is one of the oldest metabolic pathways? Justify your answer. Who wants to do that one? Any thoughts? Go ahead.
Because so if it happens in all life forms, would it have to happen in like the first ones?
Okay, that's pretty much it, right? This goes behind the hypothesis of a common ancestor. Like, hey, this common ancestor used glycolysis, then all of the offspring would use glycolysis too. Human red blood cells do not perform aerobic respiration, but they do perform glycolysis. What might happen if glycolysis were blocked in red blood cells?
Die.
The red blood cells tap into other sources of free energy needed for their functions.
I die.
The only other option would be something called, ooh, no, you can't do that either.
See, we bring.
Yeah, they would die. Okay.
Okay.
Why do you think they block? Ooh, why do you think red blood cells block aerobic respiration, or they can't aerobically respire?
Because when it's in the body, isn't it blue? And then when it oxidizes, it turns red.
Not quite. What's the function of red blood cells? Does anybody know? They carry oxygen everywhere else into your body. Okay? So if their job is to carry oxygen, you don't want them using that oxygen. Does that make sense?
Yes.
All right. What's the overall result in terms of molecules produced in the breakdown of glucose by glycolysis? We get what?
2 NADPH.
2 NADPH.
And then your ones.
2
Pyruvate.
2 pyruvates.
And
4 ATP, but we use 2, so it's a net of 2. Okay? Get up. All right, any questions on that?
No.
Sweet, we've got 15 minutes left to start on 7.3. Oxidation of pyruvate and citric acid cycle. Citric acid cycle is also known as the Krebs cycle. So when you had your readings, that's what that was over, the Krebs cycle. Think about it, it's the same thing as the citric acid cycle. Doesn't it sound so much easier than the oxidation of pyruvate?
Yeah.
Yes. How did it get from?
Probably the guy that discovered it.
That's what most people use.
Yeah, a lot of them. That's why the Calvin cycle, when we go to photosynthesis, is called the Calvin cycle, because the guy named Calvin discovered it.
Calvin, that's what I'm doing.
Interesting enough, when we get to photosynthesis, the pathway of photosynthesis is the energy goes to photosystem 2, and then it goes to photosystem 1. The reason it does that is because they discovered photosystem 1 first, and they discovered photosystem 2 second. So like in the pathway that the electrons and energy flows, it doesn't go in numerical order, which is extremely infuriating. All right, this is the second step of cell respiration. Glycolysis is the first. Where does glycolysis happen? In the cytoplasm. This is going to be the Krebs cycle or the citric acid cycle, the oxidation pyruvate. Where does this one happen? The matrix of the mitochondria. Good job. So the things you want to focus on in this section is how is pyruvate, the product of glycolysis, prepared for entry into the citric acid cycle? What are the products of the citric acid cycle? Remember, citric acid cycle is the same thing as the Krebs cycle. Okay.
I put that in my title.
All right. Again, this is kind of a visual of cellular respiration as a whole. The two pyruvates go from the cytoplasm into the matrix of the mitochondria, where CO2 is released. A little bit of ATP is formed, but a lot of electron carriers are formed. Okay, that's main Krebs cycle main role, making electron carriers that can go to the electron carrier chain. All right, so again, stages of respiration: glycolysis, Krebs cycle, also known as the citric acid cycle, and electron transport chain. Remember, oxidation is what?
Adding oxidation. No, it loses electrons.
Yeah, loss of electrons. Oil rig, oxidation is loss of electrons.
Reduction is
Gain. Or you can do Leo says GER. Loss of electrons is oxidation. Gain of electrons, GER, is reduction.
What do I say, girl?
I don't know. Alright, so in eukaryotic cells, if oxygen is present, the two pyruvate molecules enter the mitochondria, where each is converted to acetyl-CoA before entering the citric acid cycle. When it is converted to acetyl-CoA, CO2 is released, it is oxidized, meaning it transfers electrons. So it's transferring electrons to NADH. It is losing these electrons because they're no longer going to be part of the pyruvate, they're going to be NADH. And then coenzyme is attached, coenzyme is attached. How come in prokaryotics, how come it just says in eukaryotic cells it goes to the mitochondria?
Because the eukaryotic don't have those membranes of their own.
Why wasn't it in prokaryotes? Yeah, so prokaryotes don't have membrane bound organelles, so they don't have mitochondria.
Oh.
Right? Okay. They also don't have what?
Nucleus.
Very good. Alright, so oxidation pyruvate, this is prepping pyruvate to go into that citric acid cycle. You input two pyruvates, 2 NAD+. What is this NAD+ going to turn into?
NADH.
NADH. It'll pick up 2 electrons each. And you also make, you add 2 coenzyme A's. The outputs is 2 carbon dioxides, 2 NADHs, and 2 acetyl-CoAs. So we start pyruvate, our carboxyl group is removed from pyruvate, releasing carbon dioxide. So if we release carbon dioxide, how many carbons in carbon dioxide?
3. 1. Sorry.
1. CO2, 1 carbon, 2 oxygens. So if we release carbon dioxide, how many carbons are we going to be left with?
2. 1. None.
Okay. You see this?
Mm-hmm.
How many carbons are we left with?
2.
2. Okay. 3 minus 1 equals 2, everybody. We have a 3 carbon molecule and we lose 1 carbon.
2.
We get 2 carbon molecule. Alright, so this is a carboxyl group. It is lost. You notice that carboxyl group has how many carbons, how many oxygens?
3. 1 carbon, 2 oxygen.
1 carbon, 2 oxygens. CO2, it gets lost, right?
It's 3 hydrogens.
Yep. NAD plus is reduced, means it gains electrons. Reduction is gain to NADH. Okay. So we lose our CO2, it gains 2 electrons. And then acetyl-CoA, the acetyl group is transferred to coenzyme A, resulting in acetyl-CoA. So this is your coenzyme, and then this is your leftover stuff after you get rid of the CO2. Okay. Make sense?
Mm-hmm.
So, oxidation of pyruvate.
Oxidation. Losing oxygen. So you have
No. Oxidation is loss of electrons.
electrons.
Okay. So, we take pyruvate, we lose CO2,
Bye-bye.
and we transfer electrons from pyruvate to NAD plus to make NADH, right?
Yeah.
This gains electrons, this loses electrons. So this is oxidized, this is reduced, and CO2 is a byproduct. And then we end up with acetyl-CoA. So input 2 pyruvates, 2 NAD pluses, 2 coenzyme A's. We get out 2 acetyl-CoAs, 2 CO2s, 2 NADH. Try to follow along. Alright, this is just a summation in the thing. Acetyl-CoAs reshuffled, 2 carbons, or 2 of them. 3 NADH that's just created. 2. Oh, this is the whole thing.
Oh, that's a lot.
Yeah, this is the whole citric acid cycle.
Crazy.
1 ATP is made per pyruvate. 1 FADH is made per pyruvate. And this occurs in the matrix of mitochondria. The oxidation of glucose produces CO2.